Hi, everyone. It's me again. I forgot my lead to this week's post and the importance of why I even wanted to bring up the little germ-free mouse in the first place. And it's so important that I'm going to add it here. And the reason is the importance of the little germ-free mouse and the placebo. And it was occurring to me as to why the so-called quote unquote experts can always claim that MAF or GCMAF is an unproven claim, has unproven claims. And it's all based on a fallacy, false assumption, begging the question, false dilemma, and erroneous suppositions. So naturally, when you are studying a food product and the health of the gut, and bringing up the little germ-free mice model, the only way you could effectively have a clinical approach is if you introduced starvation as the counter, as the placebo or the counter argument, so that you could prove that food, basically what MAF is, and GCMAF, and milk, and everything I will go on to speak of in the post. Naturally, if you're going to introduce the importance of the health of the gut, your placebo or your clinical model would have to be starvation. And the main importance of bringing up the little germ-free mouse is that little germ-free mouse did not live very long without a microbial composition in its gut. And neither did any other animal that they tried to create a germ-free model in prior to the little germ-free mouse in the late 1950s. And that included chicks and I think guinea pigs or something like that. But so naturally, it's important. It's important that whenever you are Googling, just like it's important whenever you're Googling my former site and you see my scarlet letter, that there's always a flip side to the story. And naturally, if you are going to if you're going to create an argument or a hypothesis that a healthy gut is important, food is important, nutrition is important, naturally you're going to compare that against a germ-free model or starvation. So of course, when it comes to humans, that is not ethically feasible. So this is how they can create their fallacy argument perpetually, that there's little clinical trials or clinical support for MAF or GCMAF in humans, because you simply cannot do it in humans because it's not ethical. Because you could not wipe out a human gut, just like you really can't wipe out an animal gut and expect them to live very long. And you also can't compare starvation with nutrition. So whenever you decide to start researching this for yourself, keep this fallacy in mind, and it's one they rely on to prevent funding for this type of research. So that was my lead. That was the reason for the little germ-free mouse story and the importance in the germ-free mouse model in helping us understand everything. I then went on to explain in the post how um, your gut is 75% of your immune system, but the other 25% also wouldn't function very well without a healthy gut. Okay, so I'm going to add this to the beginning. And again, thank you for your time, and I'll see you next time. Bye. Hi, everyone. Welcome to Naturally Immune for Life. I hope you're all doing well today. And thank you for stopping by, and thank you for your time. Today, we're going to be talking about uh, the gut and the major role that it plays in your health. And most of the time, it's said that the gut is 75% of immunity. But today, I'm going to show you that that is more like 100%. Now, naturally, if you think 75%, what about the other 25%? And correctly, you would be probably thinking of your blood and your systemic immune system. But I'm going to show you that thanks to the little germ-free mice model, which was developed in the late 1950s in Notre Dame, we've learned a lot more about how the gut health affects the bone marrow and stem cells and whole body immunity. So it's often said that about 75% of your immune system resides in your gut, a shorthand reference to the enormous amount of immune tissue within the gastrointestinal tract and the gut-associated lymphoid tissue that helps detect and respond to pathogens. But the gut doesn't just provide local immunity, it has far-reaching effects on the other 25% of immune function, including immune development in the bone marrow, regulation of hematopoietic, hematopoietic blood-forming cells, and the systemic immune responses. I just had that word on the tip of my tongue, and now I forgot how to pronounce it. This broader role in the gut and immunity is now supported by numerous index studies showing how microbial signals, metabolites, and immune crosstalk influence cells and tissues throughout the body, including hematopoiesis, adaptive immunity, and systemic inflammation. First, the gut microbiota acts as a major educator and regulator of systemic immunity. While the gut and lymphoid tissue contains large numbers of immune cells that defend against ingested pathogens, microbes and their components also move beyond the gut lining into the bloodstream in small amounts. These microbial patterns and metabolites can interact with immune receptors on distant tissues, including the bone marrow, spleen, and lymph nodes. For example, microbe-associated molecular patterns such as LPS, lipopolysaccharide, and peptidoglycan can cross the gut barrier and activate innate immune sensors like toll-like receptors that influence immune cell function systemically. And I remember when I first began GCMAF, one of the first places I felt it was in my spleen. And I know that's kind of hard to believe, but I remember thinking, why does I can't remember exactly where it is right now, but I was feeling it, and I was like, what is going on? And here it does activate blood cells in your spleen. And it was like, apparently mine had been dormant or something for quite some time. So, for example, okay, yes. For example, the molecular patterns influence immune cell function systemically. Second, the microbiota plays a critical role in regulating hematopoiesis, the process by which hematopoietic poetic stem cells, HSCs, in the bone marrow differentiate into immune cells. In germ-free animals that lack a normal gut microbiota, researchers have observed defects in bone marrow populations, including reduced hematopoietic and progenitor cells and impaired development of monocytes, macrophages, and neutrophils. These defects can be rescued by reintroducing microbial signals, showing that microbial cues are essential for normal immune cell generation. So thank you to the little germ-free mouse who has been so instrumental in helping us learn so many things about the gut-brain connection as well as the gut-immune connection. Recent work has clarified that the gut communicates with bone marrow in multiple interconnected ways. Microbial metabolites, such as short-chain fatty acids, produced by bacterial fermentation of dietary fiber, enter the circulation and can modulate cytokine production and signaling pathways that guide hematopoietic stem cell fate. Short-chain fatty acids and other small molecules influence levels of hematopoietic growth factors like stem cell factor and thrombopoietin, and FLT ligand, which help maintain and expand stem cell pools. The gut's influence on the bone marrow is not only biochemical, but also involves inflammatory signaling and stromal cell interactions. Signals triggered in the gut during infection or inflammation, such as pro-inflammatory cytokines, can travel through the blood and activate bone marrow stromal cells, which in turn produce factors that stimulate emergency myelopoiesis, rapid production of innate immune cells, or maintain homeostatic hematopoiesis. These signals help the body rapidly replenish immune cells during infection. And importantly, the gut-bone marrow axis also contributes to adaptive immunity, shaping T cell and B cell functions. Microbial metabolites and structural molecules influence T cell differentiation, helping balance regulatory and effector T cell subsets that are crucial for systemic immune tolerance and pathogen defense. And I'm also going to put in here probably how milk plays a crucial role in that cell signaling. As a mammal, that is how your adaptive immunity gets developed. And it's also a preventive measure against autoimmune conditions. Changes in microbial composition, dysbiosis, are associated with altered T cell responses and with autoimmune and inflammatory diseases, indicating that the gut-derived signals impact immune regulation far beyond the intestinal mucosa. And that was just at one time just a hypothesis about 9 short years ago. And today it's common knowledge. Moreover, clinical observations show that disruption of the gut microbiota, such as from antibiotics or chemotherapy, can affect immune cell recovery and after hematopoietic stem cell transplantation, highlighting the functional significance of gut signals in human immune regeneration and disease outcomes. In summary, while a substantial portion of immune cells is concentrated in the gut, the influence of the gut microbiota on the immune system extends throughout the body through direct translocation of microbial components, production of systemic metabolites, modulation of inflammatory signaling, and regulation of hematopoiesis hematopoiesis, hematopoiesis in the bone marrow. Sorry. The gut supports the development, maintenance, and function of immune cells that reside far from the intestinal tract. This integrated network demonstrates that gut health is not just a local defense, but a central hub for systemic immune support, bridging the gut with bone marrow stem cell activity and whole body immunity. What is hematopoiesis? Hematopoiesis simply means the process by which the body makes blood cells. Hematopoiesis is your body's blood cell factory system. It happens mainly in the bone marrow, the soft tissue inside your bones, starting from special parent cells called stem cells. The body continuously produces all the different types of blood cells you need to survive. Red blood cells, which carry oxygen, white blood cells, which fight infections and support immunity, and platelets, which help blood clots and stop bleeding. Hematopoiesis is happening all the time because blood cells wear out and must be replaced. Blood cells live about 120 days, while many immune cells live only hours or days. Your bone marrow constantly adjusts production based on your needs, making more immune cells during infection or more red blood cells after blood loss. Hematopoiesis, finally, is how your body keeps a fresh, healthy supply of blood and immune cells. Finally, sorry for that constant mispronunciation of hematopoiesis. I should have said it like 10 times before I started. So that answers the question of what about the other 25% of your immune system? And it turns out the gut is just as important there as it is for nutrition and everything else. Digestion, brain health, and of course, immune health. So I'm going to add that little clip about the milk and the cell signaling. And don't forget to drink your milk, and hopefully this will help you recognize throughout the rest of the winter the importance of keeping your immune system healthy. And of course, you can learn how to do that completely on Naturally Immune for Life. And according to reports, the flu season is especially virulent this year, so you might want to keep that in mind so you don't come down with it. Okay, everyone, thank you so much for your time, and I'll see you next time. Bye.